Gaussian graphical models are employed to extract conditional dependencies between features in a dataset, often relying on the assumption of sample independence, which is rarely satisfied in reality. Existing approaches that avoid this assumption are not scalable, with cubic runtime and quadratic space complexity. This paper proposes a novel method that achieves quadratic runtime and linear space complexity without assuming independence. The proposed method is validated on both synthetic and real-world datasets, demonstrating comparable accuracy to prior work. Notably, the approach is capable of handling unprecedentedly large datasets, such as a 1 million cell scRNA-seq dataset, which was previously impossible with existing methods. The method maintains the flexibility of prior work, including the ability to handle multi-modal tensor-variate datasets and data with arbitrary marginal distributions. A significant advantage of the proposed method is that the hyperparameters are easily interpretable, unlike prior work. The authors motivate the need for scalable methods that avoid independence assumptions by highlighting the importance of conditional dependency graphs in various applications, including gene regulatory networks, tumor microenvironment structure, social networks, and clustering algorithms. The focus of this paper is on addressing the challenge of avoiding independence assumptions while preserving scalability, with a particular emphasis on the case of single-cell transcriptomics. The authors, Andrew, Westhead, and Cutillo, propose a framework for modeling conditional dependencies in single-cell RNA sequencing, scRNA-seq, data. This framework represents scRNA-seq data as a matrix with cells as rows and genes as columns, highlighting the need for scalable methods due to the exponentially increasing number of cells in datasets. Conditional dependence is defined as a measure of statistical dependence between two variables, x and y. Once conditioned over other features in the dataset, f formally, x is conditionally independent of y if the probability distribution of x and y, given f, factorizes into the product of the probability distributions of x and y, given f. The graph of conditional dependencies connects x and y if and only if they are not conditionally independent, capturing the direct effect of one variable on another. The authors focus on the case where f is the set of features with x and y removed. For normally distributed data, the authors establish a relationship between conditional dependencies and the inverse covariance matrix, or precision matrix, psi. Specifically, x and y are conditionally independent if and only if the x, y, th entry of psi is 0. The authors model the dataset, D, as a normal distribution with mean micro and precision matrix psi. However, they note that the I-D assumption may not hold in practice. To address this, they vectorize the dataset, allowing for any element in D to be connected to any other element, resulting in a precision matrix with O(n)2 square meters parameters. This formulation makes no independence assumptions, but it is computationally intractable to solve for psi. The author's approach aims to capture the complex relationships between genes and cells in scRNA-seq data, moving beyond simple correlation analysis to identify direct effects and indirect effects that pass through other variables. The paper presents a scalable multi-axis Gaussian graphical model for tensor-variate datasets, addressing the challenge of estimating factor matrices without constructing the full precision matrix. It introduces a decomposition method based on generative models, utilizing second-order edge relations and compared to other methods in Table 1. The focus is on gene and cell dependencies, assuming positive semi-definite factor matrices for axis-wise precision matrices. The graphical lasso is mentioned as a related work, addressing the optimization problem for graph inference with independence assumptions. The text discusses the need for graph factorization methods to remove independence assumptions in a computationally tractable manner. It highlights the Kronecker sum as a preferred method due to its correspondence with the maximum entropy distribution and its ability to abide by the example given in Figure 1. The original Big Lasso method had a non-optimal space complexity, leading to its unusability on large problems. Later works, such as SCBIG Lasso and IG Lasso, improved the runtime and space complexity. The Terra Lasso method extended the problem to tensor-variate datasets. The author's previous work, GMGM, focused on removing the requirement for an eigendecomposition and generalized the problem to work with multimodality datasets. However, GMGM still faced memory constraints. The text also mentions other methods that work with multimodal datasets but make the independence assumption. The authors are primarily interested in applying these methods to omics data, which is increasingly multimodal. They improved the GMGM method to have quadratic runtime and linear memory usage, enabling the use of these methods on datasets with millions of cells and/or features. The authors introduce a scalable multi-axis Gaussian graphical model, referred to as GMGM, which enhances the flexibility in handling sparse data and multimodality datasets compared to existing Gaussian graphical models. This model maintains compatibility with the non-paranormal skeptic, is extendable to multimodality and tensor-variate datasets, and can handle sparse data without densification. The authors validate their claims using both synthetic and real data, proving the existence and uniqueness up to identifiability of the resultant graphs. The model's hyperparameters are interpretable, with simple a priori choices available. Additionally, an expression for the Fisher information matrix is provided, enabling statistical hypothesis tests on the edges of the graph. The notation used primarily follows Kolda and Bader, 2009, and prior multi-axis work, with simplifications made for sanity and consistency. The multimodality Kroneker's unstructured normal distribution is characterized by sufficient statistics, whose rank is intimately tied to the Kroneker sum of matrices. To facilitate this analysis, specific notations are introduced for matrices and vectors, including the identity matrix I, the matrix of ones J, and the vector of zeros 0, which play critical roles in the context of the Kroneker sum. The derivation of the maximum likelihood estimator, MLE, requires additional assumptions, building on prior work that leverages the Gaussian copula assumption. A crucial aspect of this research is the computational complexity, which underscores the limitations of previous methods and the challenges of achieving scalability without sacrificing efficiency. The Kroneker sum structure enables a more efficient computation of the MLE, but its scalability remains a significant hurdle. The paper's contributions are thus centered around developing novel methods that can efficiently handle large datasets, thereby overcoming the limitations of existing approaches. The proposed assumptions for the scalable multi-axis Gaussian graphical model involve two key concepts: low-rank matrices for datasets and graphs, and linear sparsity. The first assumption enables the avoidance of full eigendecompositions, while the second formalizes the use of the L1 penalty by assuming graphs with DL vertices have O, DL, edges. This is in contrast to previous algorithms that make informal sparsity assumptions. The low-rank approximation is crucial, especially when dealing with singular probability distributions. The model addresses issues with singular matrices by introducing the concept of the pseudodeterminant and modifying the normalizing constant to accommodate the lower dimensional subspace. This adaptation enables the definition of a density for the singular normal distribution, which is essential for the methodology. The novel distribution, singular Kroneker's unstructured normal distribution, is particularly useful for multimodal datasets. It's defined by the product of ranks from all axes occurring in each modality. The authors introduce a method for deriving this distribution, utilizing lemmas about the stride-wise block-wise trace. This method works with tensors without repeated axes, assuming shared axes are allowed. This setup is beneficial for finding graphs in the data, especially when the data already has a graph structure. The authors introduce a novel approach to the Gaussian graphical model, focusing on scalability and multi-axis properties. They begin by presenting a generalized version of lemmas from previous research, specifically an extraction property. This property is a modified version of lemma 2 from Dahl et al., 2013, tailored for the tensor-variate case and allowing for distinct matrices X and Y. The authors provide a detailed proof of the extraction property, closely following the original by Dahl et al., 2013. The main differences lie in the tensor-variate nature of the proof and the lack of constraints on X and Y. The proof involves a series of transformations, ultimately leading to the result that the trace of the product of certain matrices is equal to the product of traces of these matrices, under specific conditions. Following this, the authors introduce the downsampling property of the stride-wise block-wise trace. This property is crucial for working with rectangular matrices in the stride-wise block-wise trace, such as those resulting from partial eigendecomposition. The downsampling property is introduced, establishing an equivalence between two matrix products. This property is crucial for deriving the method, particularly in formulating the negative log likelihood of the singular Kroneker's unstructured normal distribution. The NLL is composed of two terms: the trace of a matrix product and the log determinant of a matrix. The section proceeds to discuss maximum likelihood estimation of eigenvectors, emphasizing the importance of differentiating the log pseudodeterminant. This process is guided by Theorem 2.15 of Holbrook, 2018, and utilizes the downsampling property. The section concludes with a proof related to the symmetry of the matrix Ψ-L, highlighting its significance in deriving the MLE eigenvectors. In Gaussian graphical models, a scalable multi-axis approach is proposed to efficiently compute the maximum likelihood estimator, MLE, for model parameters. The challenge lies in computing the gradient of the negative log likelihood, NLL, with respect to the model parameters, involving complex matrix operations and trace calculations. The authors derive the gradient of the NLL with respect to the parameter Ψ-L, considering the model structure and constraints imposed by the Gaussian graphical model. This involves computing the symmetric part of a matrix expression, including terms related to the observed data, the precision matrix, and the parameter Ψ-L itself. Utilizing the definition of the stride-wise block-wise trace and leveraging properties of matrix inversion and eigendecomposition, the authors simplify this expression. By setting the gradient to 0, they obtain an equation for the MLE of Ψ-L, which involves solving a system of linear equations involving the observed data, the precision matrix, and the parameter Ψ-L. A theorem is provided stating that if the Rankel partial eigendecomposition of the observed data matrix SL is known, then the eigenvectors of SL are also eigenvectors of Ψ-L. This result can be used to further simplify the computation of the MLE and improve the scalability of the approach for large datasets. Overall, this page presents a detailed derivation of the gradient of the NLL and its application in computing the MLE for a Gaussian graphical model, designed to be scalable and efficient for large datasets and complex models. The proof by Andrew, Westhead, and Cutillo establishes the validity of using partial eigendecompositions. They introduce V gamma less than L and V gamma greater than L matrices for downsampling the stride-wise block-wise trace. This allows them to utilize lemma 2 and 3 to derive an expression for SL. The proof leverages SB trace properties to simplify the expression, resulting in a partial eigendecomposition of SL. The authors connect their method to principal component analysis, PCA, noting similar techniques can determine the optimal KL. They also discuss implications of KL being less than SL's rank, leading to the non-existence of the maximum likelihood estimator, MLE. A scalable multi-axis Gaussian graphical model can be approximated by a low-rank matrix, enabling efficient analysis of high-dimensional data. The existence and uniqueness of the maximum likelihood estimator, MLE, are crucial in this context. To address this, the authors propose a partial eigendecomposition of the matrix, focusing on eigenvectors corresponding to non-zero eigenvalues. This approach facilitates the derivation of the MLE conditions. Theorem 2 is a key contribution, describing the gradient of the negative log likelihood with respect to the eigenvalues. The proof leverages the functional invariance of the MLE, transforming the problem from finding the zero derivative of the negative log likelihood with respect to the original matrix to finding the zero derivative with respect to the eigenvalues. This strategy enables the derivation of the MLE conditions. By exploiting the low-rank structure of the matrix, the authors develop a scalable algorithm for computing the MLE. This approach has significant implications for large-scale Gaussian graphical models, where traditional methods are often computationally prohibitive. The proposed method provides a computationally efficient solution, making it possible to analyze complex systems with high-dimensional data. The authors of the research paper derived the maximum likelihood estimate for Ψ-L, leveraging theorems 1 and 2. They demonstrate that Ψ-L can be expressed as V, KL, L lambda, KL, LV, KL, TL, where V, KL, represents the eigenvectors of SL and Psi-L at the maximum likelihood estimate. The authors then address the issue of identifiability in the Kronecker sum decomposition of LL Psi-L, showing that the representation is non-identifiable due to a constant shift applied to the diagonals. This non-identifiability arises from the parameterization of the full precision matrix, which forces the inability to estimate the diagonals. To resolve this issue, the authors prove lemma 4, stating that the only source of non-identifiability in the model is the diagonals of Psi-L. This proof involves considering the last axis in gamma, denoted as passage, and showing that AUG equals ML element of GP's L equals ID gamma, gamma circled times passage plus XL element of G equals gamma ID gamma less than L circled times Psi L circled times ID gamma greater than L. This result ensures that there is no other source of non-identifiability beyond the constant shift applied to the diagonals. A scalable multi-axis Gaussian graphical model is presented, focusing on the parameterization of the precision matrix. The precision matrix is represented as a sum of Kronecker products involving the identity matrix and the precision matrix of each modality. This formulation yields off-diagonal elements directly determined by the elements of the precision matrix, while the diagonals remain non-identifiable due to the presence of the identity matrix. Notably, the non-identifiability of the diagonals does not severely impact the model, as the primary interest lies in the graph of conditional dependencies rather than the precision matrix itself. Furthermore, the gradient only depends on the identifiable part of the precision matrix, ensuring that non-identifiability does not affect the optimization procedure. In the single modality case, an identifiable representation of the precision matrix can be derived from the standard parameterization. However, in the multimodality case, identifiability becomes more complex. The authors provide examples where the traces of the precision matrices can be inferred based on interactions between different modalities. Ultimately, the traces of the precision matrices are always a linear combination of the traces of the individual modalities, highlighting the model's scalability and flexibility in handling multi-axis data. In the context of latent factor models, the matrix M-gamma plays a crucial role in establishing the relationship between identifiable quantities and true latent factors. Specifically, M-gamma determines the output space I and input space L, bridging the gap between these spaces through latent factors. The properties of M-gamma are instrumental in shaping the model, particularly the non-identifiabilities and the basis vectors of the row space R, which serve as parameters. The probability distribution function is intimately tied to M-gamma, with calculations involving the matrix's properties. Mathematically, this relationship can be expressed as insert equation, where briefly explain the variables and their significance. This expression underscores the importance of M-gamma in capturing the underlying structure of the data. Practically, M-gamma has far-reaching implications for various datasets, as demonstrated in Table 2. While the matrix offers valuable insights, its limitations must be acknowledged, particularly in scenarios where briefly mention specific challenges or limitations. Despite these limitations, the significance of M-gamma in latent factor models cannot be overstated, and its continued exploration holds promise for advancing our understanding of complex data relationships. Scalable multi-axis Gaussian graphical models, parameterization analysis. This analysis focuses on the parameterization of dataset M-gamma using basis vectors, specifically exploring scenarios for identifying parametrizations in matrix-variate, tensor-variate, and multimodality matrix-variate datasets with a consistently shared axis. The first scenario involves a matrix-variate dataset, where basis vectors are derived from the dataset itself. Each element of the basis vector is a function of the dataset elements, as demonstrated by the equations for gamma 1, gamma 2, and gamma 3, representing different sets of variables within the dataset. The corresponding parameterization matrix is provided, showcasing how the parameters are organized in this case. In the second scenario, the authors consider a tensor-variate dataset, where basis vectors are constructed by combining elements from different dimensions of the tensor. This is illustrated through the equations for gamma 1, gamma 2, and gamma 3, which now involve products of elements from different dimensions. The parameterization matrix for this scenario is also given, highlighting the structure of the parameters in a tensor-variate dataset. The third scenario deals with a multimodality matrix-variate dataset, where there is a consistently shared axis among the matrices. In this case, the basis vectors are derived by combining elements from the shared axis and the unique axes of each matrix. The equations for gamma-1, gamma-2, and gamma-3 illustrate this process, and the parameterization matrix is provided to demonstrate the organization of the parameters in this scenario. Table 2 summarizes the identifiable parametrizations for these datasets, listing the basis vectors and the corresponding parameters. This table serves as a reference for researchers working with these types of datasets, providing a quick overview of the possible parametrizations and their implications for model construction and inference. The removal of the constant k-gamma for all log 2 pi term from the negative log likelihood has no impact on the minimum or derivatives. This parameterization choice does not affect the graph of conditional dependencies or the algorithm that finds them. The authors prove the convexity of the negative log likelihood function, demonstrating a unique minimum up to non-identifiability. This is achieved through lemmas 5 and 6, which establish the convexity of the objective and the existence of a minimizer, respectively. These findings are crucial for the derivation of a statistical hypothesis test in section 2.5. The research paper discusses a scalable multi-axis Gaussian graphical model, providing a proof that the minimum to a continuous function must exist on a compact subset of the extended reals. This global minimum is ensured by convexity. An upper bound is established to rule out the case where the minimum exists only in the limit. The minimum satisfies a specific formula elementwise, involving traces of matrices and Kronecker products. This leads to the conclusion that every element of E, KL, has a corresponding element of lambda, KL, appearing in all summands, and vice versa. The authors then suppose that lambda Li equals P gamma D gamma, LLE for a given L and all I. They demonstrate that if other lambda L were 0, the sum of these lambda L terms would not add up to LE due to the lower bound of lambda, KL, being greater than or equal to EL. As lambda Li increases above this bound, the problem becomes more severe, as indicated by the gradient, which shows that the negative log likelihood increases. This implies that the minimum must occur between P gamma D gamma, LLE and EL. The assumption that E, KL, is strictly positive is necessary, as it could only be 0 in the limit as the numbers go to infinity. The authors, Andrew, Westhead, and Cutillo, investigate the issue of non-identifiability in the Kronecker product and Sylvester sum methods. They question whether an alternative choice of factorization could avoid these issues. However, it is established that both methods are non-identifiable at the diagonals. The paper then explores the problem of multiple minima due to non-identifiability. It is proposed that all alternative minima are related by the non-identifiabilities induced by the choice of factorization over the domain of rank positive semi-definite matrices. This is proven through a series of steps involving the transformation of the negative log likelihood function, which ultimately leads to the conclusion that the function is strictly convex over the domain of Kronecker sum-decomposable matrices. The paper also discusses the existence, uniqueness, and identifiability results, culminating in Theorem 3. This theorem states that there exists a unique MLE, up to the non-identifiability of the diagonals of the Kronecker sum, that estimates the precision matrix of the singular Kronecker's unstructured normal distribution. This is provided each E, KL, contains only strictly positive eigenvalues for each L, and there exists a strictly positive constant EL such that lambda, KL, is greater than or equal to EL for every L. The authors present a proof for the scalable multi-axis Gaussian graphical model, which directly follows from lemmas 5, 6, and 7. They introduce corollary 5, stating that by selecting the number of components to keep, KL, according to the formula KL equals min, rank, SL, rank, psi L, a unique solution is guaranteed, up to identifiability. The authors then shift their focus to hypothesis testing, deriving a formula for the p-value of each edge in the estimated graph under the null hypothesis of an empty graph. This is achieved by leveraging the asymptotic normality of the maximum likelihood estimator, MLE, governed by the Wald test. The MLE's error is approximated by a normal distribution, with a mean of 0 and a variance-covariance matrix given by the inverse of the Fisher information matrix, F. The Fisher information matrix is crucial, capturing relationships between the model's parameters, including crosslinks between axes and links between axes and trace terms. The authors detail the complexity of F, involving intraxis, interaxis, intertrace, and trace axis derivatives. Outlined in Appendix A, they note that F's calculation is a multi-step process, requiring careful consideration of the identifiable representation of parameters, rather than the standard representation. The negative log likelihood of the identifiable representation is provided for convenience. The total number of parameters in the model is discussed, being the sum of trace parameters and parameters per axis, given by d squared LDL, 2 plus DL1. The authors, Andrew, Westhead, and Cutillo, propose a statistical test for determining the significance of edges in a graphical model. They define the null hypothesis as off-diagonal elements of psi L being 0 and assume an average variance of Sg squared for each modality gamma. The test statistic F is derived under this hypothesis, with its calculation deferred to the appendix. The authors provide a lemma detailing the values of F for various interactions between parameters. Specifically, they outline the interactions between diagonal and off-diagonal elements of psi L, as well as those involving the parameter t. These interactions are crucial for understanding the structure of the graphical model and identifying significant edges. In Table 3, the authors summarize the allowed interactions between parameters and F. This table indicates that off-diagonal elements psi Lig do not interact with each other, while diagonal elements psi Li can interact with off-diagonal elements but not with other diagonal elements. The parameter t can interact with both diagonal and off-diagonal elements of psi L. Overall, this page discusses the development of a statistical test for edge significance in graphical models, focusing on the calculation of the test statistic F and its interactions with different parameters. The author's work contributes to the understanding of graphical model structure and the identification of statistically significant edges. A scalable multi-axis Gaussian graphical model is presented, focusing on interactions between terms in the matrix F. The model utilizes delta terms to determine interactions, which are only non-zero for diagonal elements or when j equals j I for off-diagonal elements. This leads to a block diagonal structure for F, with off-diagonal terms represented by a diagonal matrix B. The model is demonstrated using a dataset gamma 1 equals L1, L2, gamma 2 equals L1, L3, where DL1 equals 3 and DL2 equals DL3 equals 2. In this case, featuring T is proportional to the identity matrix, and A gamma equals D gamma for all, Sg, 4/2. The matrix can be expressed with exact derivations left as an exercise to the reader. A hypothesis test is defined under the null hypothesis Aug equals Sg, 2ID gamma for all, resulting in a distribution where each sample is independent. Sp gamma, L element of gamma D gamma for all. Sg, 4 per deciliter psi Lig 2, tilde operator n. 0, 1, 23. This model provides a scalable approach to understanding interactions within Gaussian graphical models and has implications for statistical inference and hypothesis testing. The paper examines the properties of edge-edge interactions in a diagonal matrix, particularly within the context of form. It highlights how inversion is simplified due to the absence of cross terms connecting edges to diagonal and t parameters. The edge-edge precision term for axis ln F can be expressed asp gamma, L element of gamma D gamma for all. Sg, 4, which simplifies to 4 DLP gamma, L element of gamma D gamma for all. Sg, 4 in the inverted form. The null hypothesis distribution for an edge estimate is discussed, showing that under certain conditions, the distribution follows a standard normal distribution. The paper also emphasizes the importance of standardization methods, such as the non-paranormal skeptic, to ensure unit variance along each axis. This is crucial for the validity of the null hypothesis and the precision matrix. The discussion concludes by noting the limitations of the test, suggesting thresholding may be preferred in certain situations. The scalable multi-axis Gaussian graphical model's practical implementation is detailed, focusing on the improved GMGM algorithm. This algorithm involves initializing variables, calculating the effective gram matrices, and iteratively updating parameters until convergence. The process utilizes theorems 1 and 2 as its foundation. The implementation is showcased in Python, leveraging libraries such as NumPy, Numba, SciPy, and Dask. The core involves calculating the effective gram matrices, partial eigendecomposition, and the iterative update process, culminating in the estimation of the solution through eigenrecomposition. The singular value decomposition, SVD, of a large, sparse dataset poses significant computational challenges. To mitigate these issues, we propose a threshold-based approach that leverages eigenrecomposition and thresholding. This method reduces the memory cost from O(PLDL) to O(NL) or O(NLDL), depending on the chosen thresholding method, and has a computational complexity of O(PLD2 lonal). An alternative strategy involves calculating the SVD directly from the data by viewing the data matrix as a gram matrix. This partial SVD approach allows for the calculation of eigenvectors directly from the input data without intermediate products. However, it requires densification of the data, which can be costly for large datasets. To address this limitation, we suggest using COCA for non-paranormal eigenvectors. This involves ranking the data and then calculating the left singular vectors of the new dataset. Although this approach requires additional space and time due to the ranking process, it provides an efficient solution for handling large, sparse datasets. In sparse count-variate data, such as scRNA-seq data, the minimum value is always 0, which is mapped to the left tail of the Gaussian distribution. This leads to issues when applying the Gaussian graphical model, as the 0 values may not be exactly centered in the distribution. To illustrate this problem, consider the cumulative density function of the normal distribution, phi. The ranking process can result in non-zero values being mapped to the center of the distribution, rather than the 0 values. To avoid this densification issue, the authors propose a transformation trick. Recognizing that all 0 values in each row get mapped to the same final value, the normally distributed value that the zeros in each row get mapped to is represented by z. The transformed data can be expressed as the sum of a matrix with the same sparsity structure as the original data and a rank 1 matrix. This approach enables the computation of the SVD of the sum of the two matrices using a rank 1 update algorithm, without densifying the input matrix. This is crucial for scalability, as it allows the algorithm to handle very large sparse datasets that would otherwise exceed 30 GB of memory. The authors also discuss the decision of which edges of psi-L to keep, proposing two methods: keeping the NL largest edges per vertex or the NL largest edges overall. The choice of method is found to be important, and its implications are explored further in the paper. In the context of sparse graph inference, a method proposed by Andrew, Westhead, and Cutillo often focuses too much on a subset of vertices, which can be detrimental to others. This issue can be addressed by downweighting edges of vertices with high degree. The choice of thresholding depends on the problem. For instance, in a graph of cells from a transcriptomics dataset, it is desirable to cluster them and assign cell types to the clusters without having many singletons. Thresholding can lead to many singletons, so keeping a minimum number of edges for each vertex may be beneficial. The authors found that overall thresholding, downweighting high-degree vertices, and keeping a small number of minimum edges performed best on scRNA-seq datasets. Alternatively, parameters can be chosen based on statistical significance, although the test suffers from low power. Ensuring the constraints required by Theorem 3 to guarantee existence of a solution is also crucial, particularly ensuring that rank psi-L less than KL. If this is consistently not met, it indicates that the chosen KL needs to be smaller. The algorithm's iterative portion takes negligible time on large samples, and there are three main hyperparameters to consider: how much to threshold, how many components to keep, and whether or not to use the non-paranormal skeptic. The non-paranormal skeptic performs well and allows the method to be effectively used on non-normal data. However, it may not be necessary for very large problems due to its need to create an intermediate sparse matrix of similar size to the input. A good a priori choice for thresholding is to keep only the edges that are statistically significant, p equals 0.05, after the Bonferroni correction. However, the test has low power, and when no edges are significant, other thresholding methods should be considered. Overall thresholding, downweighting vertices of high degree, tends to perform well, but it is reasonable to try a range of techniques. Keeping on average 10 edges per vertex resulted in good performance when used for clustering on scRNA-seq data. When the graph is only wanted as a preprocessing step, it may be reasonable to also keep a minimum number of edges for each vertex to ensure that every vertex can be assigned to a cluster. When the graph itself is the goal, choosing edges by their statistical significance when possible is recommended. In the scalable multi-axis Gaussian graphical model, the selection of the number of eigenvectors, KL, to retain is crucial. The authors emphasize that KL must be less than or equal to the rank of SL and, in practice, also less than or equal to the rank of PsiL. Notably, small values of KL can still yield good performance on real data. To determine the optimal number of eigenvalues to keep, techniques from principal component analysis, such as examining the total explained variance or scRNA plots, can be employed. The experimental setup was conducted on a MacBook Pro with an M1 chip and 8 GB RAM, utilizing Apple's Accelerate library for LAPACK and BLAS methods. The code is publicly available on GitHub and can be adapted for other architectures by using a BLAS library optimized for the user's system. In the experiments on synthetic data, ground truth graphs were generated from the Barabasi-Albert distribution using Pythonograph's implementation. This distribution was chosen due to its scale-free nature, similar to many real-world networks. Although the synthetic data may not satisfy the low-rank assumption, generating synthetic data that is both low-rank and sparse is challenging. The authors evaluate their proposed algorithm's performance using precision recall curves on synthetic data, comparing it to Terra-Lasso and GMGM. While their algorithm performs inferiorly for smaller graphs, its performance improves as the graph size increases, demonstrating promise for its intended application to large graphs with hundreds of thousands of nodes. The precision recall curves are presented in Figure 2. Precision recall curves for two Barabasi-Albert graphs of different sizes, 250 nodes and 5,000 nodes, are presented, comparing the performance of various graph embedding methods, including GMGM, GMGM 125 PCs, GMGM 60 PCs, and Terra-Lasso. Figure 2 displays the curves, with the z-axis representing recall and the y-axis representing precision. The results show that GMGM and its variants outperform Terra-Lasso in capturing the underlying structure of the graphs. In the 250-node graph, GMGM achieves the highest precision in recall, while the explained variance plot indicates that the used eigenvalues account for nearly 100% of the variance in the data, with GMGM and Terra-Lasso making no low-rank assumptions. In the larger 5,000-node graph, GMGM and its variants continue to outperform Terra-Lasso, which was omitted due to its inability to run in reasonable time. The precision recall curves for GMGM and its variants are relatively consistent across the two graph sizes, demonstrating the scalability of these methods. The shaded regions around each curve represent the maximum and minimum values reported over 10 runs, with the central curve being the mean of the maximum and minimum. These results suggest that GMGM and its variants are effective graph embedding methods that can capture the underlying structure of graphs of varying sizes, outperforming Terra-Lasso in terms of precision and recall. Scalability is a crucial aspect of multi-axis Gaussian graphical models, particularly when dealing with large graphs. As the number of nodes increases, runtime efficiency becomes a significant concern. This is exemplified by the performance of algorithms such as Terra-Lasso and Dinin-Lasso, which exhibit a trade-off between accuracy and speed. Notably, the data generation process for a 10,000-node graph takes over 7 minutes, underscoring the need for efficient methods. The generation process itself is a critical component, with computation time increasing significantly as problem sizes grow. This highlights the importance of developing SVD-free approaches capable of handling larger datasets. The current reliance on singular value decomposition, SVD, hinders scalability, making it essential to explore alternative methods. In the context of real-world data experiments, preprocessing is a crucial step. The non-paranormal skeptic is employed to prepare the data, ensuring that it meets the necessary assumptions for the model. Subsequently, the Bonferroni correction is applied to account for multiple hypothesis testing and ensure statistical significance. Ultimately, the development of efficient and scalable algorithms is crucial for the widespread adoption of multi-axis Gaussian graphical models in real-world applications. By addressing the current limitations and exploring novel approaches, researchers can unlock the full potential of these models. The GMGM algorithm is a method for computing the precision matrix of a Gaussian graphical model, comprising three main components: eigenvector calculation, eigenvalue calculation, and recomposition to produce the final precision matrix. As shown in Figure 4, eigenvector calculation dominates the total computation time. The authors apply the GMGM algorithm to reconstruct the frame graph of coiled 20 videos, comparing its performance with other methods. Table 4 presents the results, categorizing connections based on their distance in the video sequence. This analysis provides insights into the accuracy and efficiency of each method in identifying correct edges and connections between frames. The comparison includes GMGM with 10 principal components, and Terra-Lasso. The GMGM algorithm with 10 principal components aims to reduce the dimensionality of the data, thereby improving computational efficiency. The Terra-Lasso method, on the other hand, is a scalable algorithm for learning Gaussian graphical models. The results in Table 4 demonstrate the effectiveness of the GMGM algorithm in reconstructing the frame graph, with improved accuracy and efficiency in identifying correct connections between frames. The author's analysis highlights the significance of the GMGM algorithm in computing precision matrices for Gaussian graphical models, particularly in applications involving high-dimensional data. In the context of multi-axis Gaussian graphical models, the authors conduct a comparative analysis of various methods for learning the frame graph. The methods under investigation include the proposed GMGM method, GMGM with 10 principal components, GMGM with 10 principal components and 1 modality, and Terra-Lasso with 1 modality. A visualization of the comparison is provided in Figure 5, which displays the connection's precision and recall for each method. The results are further detailed in Table 5, which reports the precision and recall of each algorithm for different edge tolerances, specifically exact, 1 off, 2 off, and 3 off. The table highlights the superior performance of the GMGM method, achieving the highest precision and recall across all tolerances. The authors then apply their method to the coiled 20 video dataset, a standard testbed for multi-axis models. This dataset consists of 20 videos, each featuring a simple object rotating 360 degrees, with 72 frames and 128 by 128 pixels. The frame graph is expected to exhibit a circular structure, which the authors model using a singular frame graph and individual row and column graphs for each object. In their experiments, the authors retain only the edges that are statistically significant to the 5% level after applying the Bonferroni correction. They report results using all eigenvectors and the top 10 principal components. For comparison with prior work, they also run their algorithm on a single object, the duck object, and retain the same number of edges as in the prior work. The comparison of GMGM and Terra-Lasso methods on the coiled 20 dataset reveals that GMGM outperforms Terra-Lasso, particularly when considering the whole dataset. The results are presented in Figure 6, which demonstrates GMGM's superiority in object graphs produced. Moreover, the algorithm is applied to real-world data, specifically the head and neck single-cell transcriptomics dataset. Pre-processing steps include normalization, filtering, and scaling of the data. The application of the algorithm yielded a sparse gene graph with 1048 edges, highlighting the potential of GMGM for identifying meaningful relationships within complex biological systems. The GMGM method excels due to its ability to handle high-dimensional data efficiently and accurately, making it a valuable tool for researchers in the field of bioinformatics. Its performance in the coiled 20 dataset and the head and neck single-cell transcriptomics dataset showcases its capacity to identify relevant patterns and connections in large datasets. In conclusion, The GMGM method offers a promising approach for researchers seeking to analyze and understand complex biological systems through network analysis. Its superior performance compared to Terra-Lasso and its successful application to real-world data underscore its potential in advancing our understanding of gene interactions and their role in various biological processes. The development of a scalable multi-axis Gaussian graphical model is presented for analyzing gene expression data in the context of immune response and cancer research. This model is applied to a dataset of head and neck cancer patients, focusing on specific gene sets related to humoral immune response, antibacterial humoral response, peptide antigen assembly with MHC protein complex, and myeloid cell activation involved in immune response. To validate the local structure of the learned graph, the authors utilize gene ontology (GO) terms, which provide a standardized vocabulary for annotating gene products. Additionally, the Leiden clustering algorithm is employed to investigate the global structure of the gene networks. The results are compared to those obtained using the Terra-Lasso algorithm, a non-scalable method that can only be applied to smaller subsets of the data, such as the data from a single patient. The proposed multi-axis Gaussian graphical model offers a scalable solution for analyzing large gene expression datasets, enabling the identification of complex relationships between genes and their roles in immune response and cancer development. By integrating GO terms and Leiden clustering, the model provides a comprehensive framework for understanding the local and global structures of gene networks. The analysis of gene expression focuses on specific biological processes, including RNA splicing and mRNA processing, cytoplasmic translation, and peptide biosynthetic processes. P-values are provided for these processes, highlighting their significance. The immune response and immune system response are also discussed, with a focus on leukocyte activation. Additionally, the page covers cytoplasmic translation and peptide biosynthetic processes again, as well as organonitrogen compound metabolic processes, oxidative phosphorylation, and protein metabolic processes. The study utilizes algorithms and tools like G-Profiler to identify enriched GO terms, particularly in the immune response. Results are presented in a tabular format, showing p-values and gene counts for each process. The application of a scalable multi-axis Gaussian graphical model to a million-cell dataset for analyzing the humoral immune response has been explored. This algorithm significantly identifies statistically significant edges and clusters associated with immunological properties. The dataset, comprising 1,248,980 cells and 36,571 genes, was preprocessed to retain only the top 2,200 highly variable genes. The analysis focuses on the biological process GO category for the largest clusters, revealing associations with coherent communities of genes. The results demonstrate the algorithm's ability to capture processes that hold true across all patients, showcasing its potential in large-scale immunology research. This approach can provide insights into the complex interactions within the immune system and help researchers better understand the dynamics of the humoral immune response. The research paper presents the results of a clustering analysis using the GMGM algorithm on a dataset, identifying 6 clusters associated with specific biological processes and pathways. For instance, cluster 1 is linked to cell-cell adhesion, apoptosis, and positive regulation of cellular processes, with significant p-values indicating the relevance of these processes in this cluster. The pattern continues for the remaining clusters, each being associated with distinct biological processes such as immune response, leukocyte-mediated immunity, and immune system process. The authors also discuss the computational complexity of their algorithm, including time and space requirements for calculating singular vectors, performing convex optimization, and eigenrecomposition, as well as the non-paranormal skeptic method's additional complexity due to ranking the data and its sparsity considerations. The authors introduce a scalable multi-axis Gaussian graphical model for matrix-variate data, capable of handling datasets exceeding 1 million samples on a personal computer. With a constant number of latent variables, KL, and matrix dimensions, DL, where NL element of O, DL, the computational complexity of their algorithm is O, D2 plus D2 logged, in time and O, D plus SD2, in space. This significantly improves upon previous work on matrix-variate data, which is limited to unimodal matrix-variate data. The authors validate their results using synthetic data and 3 real-world datasets, supported by rigorous proofs of the existence and uniqueness of the results. The hyperparameters of their model are interpretable, with the low-rank approximation analyzable in terms of explained variance and the thresholding parameter selectable through a statistical hypothesis test derived in the paper. Future work avenues include generalizing the model to handle multi-modality datasets with partial overlap of axes and relaxing the Gaussian copula assumption. They also aim to develop an efficient eigendecomposition-free algorithm, which would enable further scalability, particularly in the presence of regularization. The Fisher information is derived in this section, omitted from the main paper due to its length and technical nature. The negative log likelihood, NLL, is defined as the sum of 3 terms: the log determinant of a matrix, the trace of the product of the inverse of this matrix and a sum of matrices, and the trace of the product of the inverse of this matrix and another matrix. The Hessian of the NLL is then derived using lemmas 9, 10, and 11. These lemmas compute the partial derivatives of the NLL with respect to various parameters, involving the trace of matrices and their products. The proofs of these lemmas are similar to those of earlier lemmas, demonstrating the technical depth and complexity of the calculations. The development of a scalable multi-axis Gaussian graphical model is a pivotal contribution to the field. It begins with the partial derivative of the negative log likelihood with respect to the model parameters, which highlights the importance of these parameters and their relationships. This is followed by the second derivative, demonstrating how the model's parameters interact and influence each other. The paper also explores the derivative of the negative log likelihood with respect to the psi-ljk parameters, showcasing the model's adaptability and flexibility. This section emphasizes the scalability and flexibility of the proposed model, making it a significant advancement in Gaussian graphical models. In this section of the research paper, the authors Andrew, Westhead, and Cutillo explore the intricacies of computing derivatives and symmetrization processes. They introduce lemmas essential for understanding the mathematical framework, starting with lemma 11, which demonstrates how to compute the unsymmetrized derivative of the negative log likelihood, NLL, with respect to the parameters of the model. This involves a sum over all possible combinations of 2 nodes in the graph, leveraging the previously established results in lemma 9. The authors then symmetrize this derivative, resulting in a more complex expression that captures interaxis connections within the model. Following this, they introduce lemma 12, which provides a general result for computing the trace of a product of matrices when some of these matrices are identity matrices. This lemma is crucial for simplifying expressions and will be used extensively in subsequent sections. Finally, corollary 9 is presented, stating that under certain conditions, the trace of the product of matrices can be computed by summing over dimensions of identity matrices and Kronecker delta terms. This result is pivotal for establishing the values of F under the null hypothesis, as outlined in lemma 8. The lemma on page 43 of the multi-axis Gaussian graphical model establishes a relationship between the null hypothesis on the variance matrix AUG and subsequent expressions for FTI, FTI psi ljk, and F psi L1I1J1 psi L2I2J2. These expressions involve the variance component, SG, 2 and the model's parameters. The proof section derives the partial derivatives of the negative log likelihood, NLL, with respect to T and psi ljk, highlighting the impact of these parameters on the model's likelihood. The algebraic manipulations demonstrate how the stride-wise block-wise trace term evolves under various transformations, showcasing the intricate relationships within the model. This section is pivotal in understanding the behavior of the model under different conditions, particularly in the context of variance and parameter estimation. Computation of psi L1I1J1, psi L2I2J2 is a crucial step in the research. This computation is performed in 2 distinct cases: when L equals L1 and L2, and when L1 does not equal L2. In both scenarios, the authors employ lemma 12 to facilitate the calculation. In the first case, where L equals L1 and L2, the non-symmetric derivative is utilized. To symmetrize this derivative, The authors average over all possible symmetries, swap indices I1 and J1, and swap indices I2 and J2. This approach enables the analysis of both same-axis and different-axis cases, revealing the impact of symmetries on the computation. The same-axis case is then rewritten, leading to a comprehensive proof. This rewritten expression is a key finding, as it provides a novel perspective on the computation of psi L1 I1 J1, psi L2 I2 J2, when L equals L1 and L2. In contrast, when L1 does not equal L2, the authors apply a distinct approach, again leveraging lemma 12. This alternative method allows for the efficient computation of psi L1 I1 J1, psi L2 I2 J2, in the case of differing L values. A scalable multi-axis Gaussian graphical model is a statistical framework designed to handle high-dimensional datasets with complex relationships between variables. This model has been applied in various fields, including genomics, where it is used to analyze gene expression data and identify co-expression networks. The Gaussian graphical model, GGM, is a probabilistic model that represents the conditional independence relationships between variables in a graph. Each node in the graph represents a variable, and the edges represent the conditional dependencies between them. The multi-axis GGM extends this framework by allowing for multiple types of edges, each representing different kinds of relationships. In the context of genomics, these edges can represent different types of interactions, such as transcriptional regulation, protein-protein interactions, or genetic correlations. By incorporating these different types of relationships into a single model, researchers can gain a more comprehensive understanding of the complex interactions within biological systems. Several algorithms have been developed to fit these models to data efficiently, including the GM-GM algorithm and TERR-Lasso. These methods are particularly useful for analyzing datasets with multiple types of relationships and high-dimensional variables. Studies have applied these models to real-world datasets, identifying conserved regulatory networks across different organisms and key immune cell types and their interactions in cancer biology. Overall, the scalable multi-axis Gaussian graphical model provides a powerful tool for analyzing complex high-dimensional datasets and identifying patterns of conditional independence. This page from the research paper provides a comprehensive overview of various algorithms, techniques, and methodologies used in machine learning, artificial intelligence, and computer science. It begins with a reference to Fang Han and Han Lu's work on high-dimensional semiparametric scale invariant principal component analysis, which was published in the IEEE Transactions on Pattern Analysis and Machine Intelligence in 2014. The authors then discuss the NumPy library, an array programming tool developed by Charles R. Harris et al., as described in their Nature article from September 2020. NumPy is a crucial library for scientific computing and machine learning tasks due to its ability to efficiently handle large, multidimensional arrays and matrices. The paper also mentions Alfredo Kalaitzis et al.'s work on the bigraphical lasso, presented at the 30th International Conference on Machine Learning in 2013. This method is designed for sparse network inference from count data, offering insights into the relationships between variables in complex systems. Additionally, the authors cite Lees Kohlberg et al.'s update on G-Profiler, a web service for functional enrichment analysis and gene identifier mapping, published in Nucleic Acids Research in July 2023. This tool is essential for researchers working with genomic data, as it facilitates the identification of biological pathways and processes associated with specific genes. Furthermore, the paper, in the realm of machine learning and artificial intelligence, several significant research papers have contributed to our understanding of complex algorithms and datasets. A pivotal work by Calvin McCarter and Si-Young Kim, published in Advances in Neural Information Processing Systems in 2014, introduced sparse Gaussian chain graph models. This model has been instrumental in understanding the relationships between variables in large datasets. Another key resource is the Columbia Object Image Library, COIL-20, a dataset designed for object recognition and image understanding. This dataset comprises images of 20 different objects, providing a comprehensive platform for researchers to test and refine their algorithms. Furthermore, Ivan Peshekhanov, Alexei Arzantsev, and Maxim Rakuba's paper on training a Tucker model with shared factors, presented at the 27th International Conference on Artificial Intelligence and Statistics in 2024, has shed light on the importance of shared factors in Tucker models. This methodology allows for more efficient data analysis and improved predictive accuracy. These resources collectively underscore the importance of advanced algorithms and datasets in advancing the field of artificial intelligence. They highlight the ongoing efforts to refine machine learning models and improve their performance, ultimately leading to more accurate predictions and enhanced decision-making capabilities. In the paper titled, Ig-Lasso, Scalable Estimation of Cartesian Product of Sparse Inverse Covariance Matrices, Jun-Ho Yoon and Si-Young Kim propose a novel method for estimating the inverse covariance matrix of a large dataset. Ig-Lasso efficiently handles high-dimensional data and sparse inverse covariance matrices, making it particularly useful in applications where the number of variables far exceeds the number of observations. The authors begin by discussing the importance of inverse covariance matrices in statistical analysis, highlighting their role in estimating the partial correlations between variables. However, they also note that traditional methods for estimating these matrices can be computationally expensive and may not perform well when dealing with sparse data. To address these limitations, the authors introduce Ig-Lasso, which combines the benefits of the graphical lasso, GLASSO, and the Cartesian product of sparse inverse covariance matrices. Ig-Lasso uses a block coordinate descent algorithm to efficiently estimate the inverse covariance matrix, taking advantage of the sparsity in the data to reduce computational complexity. The authors demonstrate the effectiveness of Ig-Lasso through a series of experiments on synthetic and real-world datasets, showing that it outperforms existing methods in terms of both accuracy and computational efficiency, particularly when dealing with large datasets.